All right, I am Melissa Smith, and I am a research ecologist at the USDA Agricultural Research Service Invasive Plant Research Laboratory in Fort Lauderdale, Florida. We look at how invasive plants affect primarily the Florida landscape and how to utilize biological control along with other methods to control large-scale invasive plants. And I came to this place through my graduate work in community ecology that focused on invasive plants in particular. And so I will be focusing today, though, on how interagency cooperation between federal, state, and local agencies helped to forward the success of invasive plant control campaign in the Florida landscape, and particularly against an aquatic wetland invader. So this is primarily a talk that revolves around people cooperating. Ecosystems don't really cooperate, they interact, but we are fortunate enough to work in an environment in Florida in which local and state agencies are readily at the same table. So this includes U.S. Fish and Wildlife Service, the USDA, Florida Fish and Wildlife Conservation Commission, the University of Florida. We work extensively in the national parks, so Everglades National Park and Big Cypress National Preserve. And then much of our support comes through a cooperation between the U.S. Army Corps of Engineers and the South Florida Water Management District. And I'll talk about that a little later in the presentation. And this is a picture of my colleagues, my technicians, and the group that does most of the work here at the Invasive Plant Research Lab. Many of these are interns, many of these are low-level technicians who gather the data and really do all of the on-the-boots work, and they rarely get recognition. So I just wanted to take a minute and really recognize the people in this picture, because they are the heart and soul of a lot of invasive plant research. So let's start with a little bit of background. South Florida, and Florida in general, exists on a peninsula that ranges from about 10 to 15 feet above sea level to right about 1 or 2 feet above sea level. And so we interact with both land and water really frequently. Historically, the chain of Kissimmee Lakes, which is north of Lake Okeechobee, flowed through a series of underground springs and rivers and lakes into one of the largest lakes in North America, Lake Okeechobee. And from there, it would go either out through the Caloosahatchee River, a little bit of westward flow, but primarily you had a north to south flow from the Kissimmee through South Florida. And that propagated a massive wetland system that included marsh ecosystems, it included deeper wetland systems, lakes, rivers, sloughs, and this historical flow is what created what we think of as the Everglades. Now, this, however, doesn't facilitate human inhabitants very well. So historically, human inhabitation consisted mostly of people living on the coasts. So this included the Tequesta, as well as some other groups that were mostly coastal peoples but would travel inland, sometimes right about here, to higher areas to do some trading. And then when the Seminole and Miccosukee came down in the early part of the 19th century, they hid in the swamps, quote unquote, in South Florida in order to escape capture. So for most of human history in South Florida, they inhabited the higher ridges on the east and west coasts rather than the interior, which left the interior open to birds and other wildlife and a wholly unique ecosystem that is a a conglomeration of subtropical temperate floras as well as wetlands. However, when Florida opened up to settlers and to larger populations, it was during the early part of the 20th century and late part of the 19th century when plumage and plumes on hats was very important and a huge economic driver for harvesting birds out of the Everglades. So this all occurs within a historical context. You can see behind the picture on the right that you've got Dade County pine as well as probably some planted maples, but you don't see the characteristic Australian pine in the background like you do now. In addition to harvesting out of the Everglades, there was a big campaign into making the interior of Florida more usable, quote unquote. This meant draining or canalizing much of the water that flowed naturally south of Lake Okeechobee in a wide swath. So that some of that land would dry up and be arable for agriculture. These were called the Central and South Florida projects, and those started in the 40s. Land reclamation started as early as the 1920s. And this was to really promote the use of Florida land for sugar and citrus and tomatoes, and to really help Florida's not only agricultural boom, but the housing boom. People wanted to live in Florida for the same reasons then as they do now. It's warm in the middle of January, and the climate during winter is very conducive to human activities. However, this increased disturbance as well as increasing plant importations. People, when we move to new places, often bring plants that are familiar with us. And we had two major importers in Florida. One was with the USDA, David Fairchild, and he had a collection center in Florida, which is now the Fairchild Botanical Garden, as well as his personal home. And he brought in a lot of different plants and plant species, some of which are now invasive. Henry Neerling also brought in a lot of plant species. He was up north of Lake Okeechobee in central Florida. And then there was also the Royal Palm Nursery. So all of these groups are bringing in plants, subtropical, tropical plants to grow and to use in the burgeoning horticulture industry. One of the plants that they brought in was punk tree or melaleuca, so Melaleuca quinquenervia. It's not the same as what you traditionally think of as a tea tree. That's a different melaleuca species. But starting in the late part of the 19th century, Royal Palm Nursery, Neerling in central Florida, and the USDA brought in many individuals of Melaleuca quinquenervia from the east coast of Australia. This importation was primarily for horticultural usage, and so we can find historical trees all throughout old neighborhoods in Florida, especially South Florida. So in my neighborhood in particular, there are two trees which, unbeknownst to my neighbors, I cored, and they are from the 1920s. So these are really old trees that people initially planted for their looks. It's a nice big tree. But it also grows rapidly under a myriad of conditions, especially in wet conditions. And so as the U.S. Army Corps of Engineers was conducting its canalization or channelizing projects in order to help make Florida more livable for both people and agriculture, they used this tree, Melaleuca, in order to shore up those levees. This is all happening, though, at the same time when folks are starting to promote the idea of conserving the wetlands in South Florida from the lake and whatever is left south. Ernest Coe, who is pictured here, was instrumental in getting the park designated. So the park was designated in 1934, and here he is sitting in one of the, cypress marshes. And then in 1947, the Everglades National Park was dedicated. However, this is long after we have gone beyond the point of altering crucial ecological processes such as water flow and hydrology, as well as importing plants that drastically change things like water flow, hydrology, and fire. Interestingly enough, pictured in the back here, you can see Australian pines, which is another invasive species we have in South Florida. And there's a big basket of citrus that shows the often conflicting interests of conservation in South Florida. We want to have agriculture and we want to promote agriculture in South Florida, but we often do so at the peril of conserving critical habitat for species of concern. One of the major champions of Everglades National Park was this woman pictured in the middle, Marjorie Stoneman Douglas. And so this illustrates at the very beginning how the establishment of parks and of conservation areas is almost entirely a union and a cooperation between federal agencies who manage and who can purchase the land, and private citizens who can champion and lobby on behalf of those places. That said, Everglades is unique because it's preserved, not because it has major geological features such as big mountains or beautiful lakes. It's preserved because of its critical habitat and function, and so it's preserved for its biology, and that is captured well in Ms. Stoneman Douglas's seminal book, The Everglades River of Grass. So as we move through the 30s and 40s, we're discovering that we want to have a larger conservation idea, but we're still bringing in plants, and that includes Melaleuca. It was planted extensively through the 40s and 50s and even into the 60s. It then got onto everybody's radar that this was perhaps a species that could self-promote, that could sustain itself, and that could increase its range throughout much of South Florida. So in the 60s and 70s and even the 80s, we had huge areas that were now being overtaken by this tree. Unfortunately, the natural processes of fire that are part of the South Florida landscape caused a lot of damage, because when burned, Melaleuca releases huge amounts of volatile organic compounds, and those can be very irritating to people that inhale the smoke. And then because Melaleuca is extremely good at uptaking water, we also experienced something we never really thought we would, which was drought. And so it compounded issues with hydrology that were already starting with moving water instead of from north to south, starting to move water from east to west out of what is a natural slough. Like most large-scale invaders, Melaleuca has a myriad of ecosystem-scale impacts, including altering water depth and surface flows, which are critical for aquifer recharge in South Florida. All of us who live down here are benefactors of the Florida aquifer, which for our drinking water, and that is provided by water that flows over the Everglades and it flows through porous limestone and into that aquifer about 1,000 feet down. It basically stopped succession in these communities. So not only did Melaleuca change the hydrology, but it also made it so that plant recruitment under a Melaleuca canopy was quite difficult. It altered light availability and nutrient availability, as well as disturbance regimes. So in the late 70s and 80s, we really started to talk about this. And that's because it was affecting massive areas of land that were not only controlled by private citizens, but also huge areas of federally owned and managed land. So the area in light gray that doesn't have an outline is what we consider historic Everglades wetlands. So these are areas that are not part of a highland hammock or anything like that, pinelands. These are areas that Melaleuca could potentially grow in. Areas that are bounded are Big Cypress National Preserve, which is this area here. To the south of that is Everglades National Park. And this area right here, which is actually maintained by a series of levees and boundaries, is Loxahatchee National Wildlife Refuge. All of these areas are what we consider the greater Everglades ecosystem, and we manage those, or we address problems on those sort of holistically. The dark area, though, is the pre-treatment Melaleuca distribution. So this is before large-scale removal efforts began in the 1990s. Just to re-emphasize not only the fact that these are federally managed lands, but you also have these water management area designated lands, which are often called storm treatment areas, storm water treatment areas, and those are managed by the state. You have tribal lands, so this here is the Miccosukee Indian Reservation. To the north of that is the Big Cypress Reservation. So all of these are stakeholders in Melaleuca control and of making sure that ecosystem processes function well. However, when we get above here, this is the entire South Lake Okeechobee agricultural area, and then we have huge agricultural areas down here in the Homestead Redlands area. So again, we have this interplay with agriculture, with large densely populated areas that are directly adjacent to massive conservation areas. So let's quickly go through Melaleuca control. We've sort of talked about the evolution of a conservation ethic in Florida, but really beginning in the 1970s, Florida Fish and Wildlife Conservation Commission started investigating herbicidal control, and that was just before the U.S. Forest Service and the Fish and Wildlife, or excuse me, South Florida Water Management District did several surveys of Melaleuca density in Florida. In 1979, the U.S. Army Corps of Engineers began doing herbicide trials on Melaleuca at Lake Okeechobee along that southern levee area. In 1982, in order to address the burgeoning exotic plant problem in Florida, the Florida Exotic Pest Plant Council, or FLEPC, was formed. In '86, the USDA Agricultural Research Service down here at the Invasive Plant Research Lab began to conduct surveys for biological control agents, primarily in its native range of Australia, but also New Caledonia. And then the South Florida Water Management District and the National Park Service started to conduct serial reconnaissance flights. So these are overhead flights to survey Melaleuca, again from the air, as well as other invasive species. In 1996, we had the formation of the South Florida Ecosystem Restoration Task Force, which then promoted and prompted the formation and the legislation that now governs the Comprehensive Everglades Restoration Plan. In '97, and I'll talk about this obviously in more detail, that's when our first biological control agents were approved for release. And in 2002, the Invasive Plant Research Lab embarked upon a multi-agency cooperation called Tame Melaleuca, or the area-wide area-wide study for Melaleuca eradication. So this idea, or excuse me, Melaleuca control, this idea was then to utilize multiple plots on multiple different types of land in order to achieve long-term control. The main goal of this, and really most of our studies, is to look at how can we integrate different management techniques in order to achieve long-term control. One technique that we looked at is mechanical control. So this is when we use bulldozers and chippers and grinders in order to destroy large trees. And sometimes when we think about, I'll just explain the quote at the top, when we think about Everglades restoration efforts, an official from the Department of Interior probably put it best, that when we're talking about restoration, an Everglades landscape teeming with exotic species is not a restored Everglades. And the effort of removing Melaleuca is there in order to reestablish better ecosystem function without this exotic species. Mechanical control can remove large trees, but it's incredibly labor intensive and expensive. It can cause non-target damage. If there are native plants in the understory, they're going to be affected and probably harmed by a chipper. It doesn't control coppicing, which is what happens when we cut the top of a tree off, but the stump is still alive, and so you get big bushy outgrowth from that stump. And then Melaleuca is a prolific seed producer, and mechanical removal does nothing for reducing seed recruitment. Most of you are familiar with chemical control, which is utilizing herbicides to get rid of invasive plants. The pros are that with certain removal techniques, we can get rid of large trees. It's very effective. Hand spraying techniques that target specific trees and specific individual trees can minimize non-target impacts. But again, it's very labor intensive, especially when you're talking about Everglades habitat. People are often walking out in wet, if not entirely inundated conditions. They're remote. They've got to get there via an airboat. If we decide that the infestation is too high, then they can utilize aerial spray, so spraying from a helicopter or a fixed-wing aircraft, but that can kill massive amounts of non-target species. And again, the seed bank of Melaleuca can be massive, and so there's no control of seedling recruitment. So you'll notice that I've mentioned seedling recruitment several times. So in our first effort to find a biological control mechanism, we often target where the other two control mechanisms are falling short. The pros of biological control in Melaleuca is that it reduces seedling recruitment. Once these insects are introduced, there's really no maintenance or follow-up costs except for the cost of studying impact. These are self-sustaining populations, and because of host range testing, we know that there's no non-target direct damage. The cons are that it's expensive to develop biological control up front, and it is just that. It is control, not eradication. So just think that these are self-perpetuating populations. They're never going to eat all of their particular host plant. They will always, keep enough for a population to go. So let's explain, for those that are not familiar with classical biological control, a little bit about how this works. So say that you have this bad plant, but again, there are no bad plants. It's just a plant that is growing where it shouldn't grow. So, but never mind the verbiage. So we will go back to where it's native. In this case, Melaleuca is native to Australia. And interestingly enough, many of our invasive plants in North America are native to either Australia or Asia. Shows that we were over there getting plants pretty frequently in the 19th and 20th centuries. So we'll survey its native range, and we focus on specific specialists, not generalists. So mammalian herbivores are often generalists, so we generally look at the insect flora. In this case, we want to make sure that it is extremely host specific, that the insects that we choose, or the arthropods, we also utilize mites, will only eat that plant and that plant alone. So occasionally you can get a little bit of test feeding on something else, but we want to make sure that reproduction and primary feeding only happen on the target species. And so these species then spend about 6 to 8 years, or excuse me, 4 to 8 years in a federal biosafety level 2 quarantine facility. So nothing that comes into this facility goes out of this facility until it has been permitted to by the Animal Plant Health Inspection Service. So APHIS gives the permit. And once it's released, though, after showing host specificity and other benchmarks, then it's approved for release, and we start mass rearing and releasing this insect. Just to sort of give you an idea of what happens in the field. So this is a field host specificity test. We released multiple individuals, multiple adults, and followed them through the top graph is adult recruitment onto plants, so whether or not adults were present on species. And then we also looked at larval recruitment, so how many days larvae, or how many larvae were found on each species throughout the year. So you'll notice, that Melaleuca quinquenervia is the square, and it's right here. We did actually get adult recruitment on a non-native species, but in larval recruitment, we get primarily all of the larval recruitment is on Melaleuca quinquenervia, and then a couple of other Melaleuca species, none of which are native to Florida. So this is a very good species in terms of its ability to specialize on Melaleuca quinquenervia and maintain those populations over time. So we utilized this integrated management approach and brought multiple stakeholders that specialize in each one of these efforts into the larger study. The U.S. Army Corps of Engineers and the South Florida Water Management District focused on mechanical removal. IFIS, so the research arm of the University of Florida, and the Florida Fish and Wildlife Conservation Commission helped with mechanical removal. And then UF, as well as the Invasive Plant Research Lab, focused on developing biological control. We had several research sites throughout South Florida and throughout the range of both the historical Everglades and Melaleuca. All of these sites had had biological control organisms at them. So they had the Melaleuca weevil, which is this very cute guy, as well as the Melaleuca psyllid. So these are sap feeders, and they produce this flocculence. So both of these are very effective and self-moving organisms. The biggest issue, though, is that these guys require soil in which to pupate. These insects do not. We had 2 control sites where we used pesticides in order to keep these insects off, to look at the effects of just mechanical or chemical removal, but every other site had some combination of chemical, mechanical, and biological control. We utilized hack and squirt, aerial sprays, chainsaws, and a feller bench, which is an attachment to a backhoe that grinds everything in its path. And we found that in permanently wet sites where the weevil can't propagate, the psyllids thrive. In places where both the psyllid and the weevil are present, we had a 99% reduction in seed production and recruitment. This is massive for controlling Melaleuca. At this point then, large trees can be treated and removed, and maintenance costs then for Melaleuca dominated lands have decreased significantly since the 90s, so over the past 20 years. And pictures always help explain things better than words. So this is Moorhaven Marsh at Lake Okeechobee. This is 1994, before treatment in earnest began. All of this green area is thick Melaleuca trees. This is from 2007. There are remnant Melaleuca trees, but each one of these can be treated and easily removed. And it's primarily native habitat now, and that is because we went in and removed these large swaths of trees because of the land area and the fact that there is some dry area. Weevils were able to be present in those, and they pretty much stopped seedling recruitment. And then the occasional spray is conducted in order to remove individual trees. So what once was a landscape level invasion is now individual trees. And this is what we're seeing throughout the landscape. So in 1995, each one of these cells that's darker colored, darker red, is a heavy infestation of Melaleuca. In 2001, we start getting major efforts to treat Melaleuca, especially on water management district land. So we still have a couple of places where we have heavy infestations, but you can see that especially down here in the southern buffer areas, we're decreasing these efforts. In 2005, same story, and in 2010, we've really managed to push Melaleuca and Melaleuca-dominated lands to specific areas that have either no treatment or that the treatment that's available is difficult because of the landscape, particularly very wet areas. More recently, we found through aerial sketch mapping, so this is where we go over each one of these squares as a grid, and we fly over them and estimate coverage. Melaleuca and the severity of the Melaleuca invasion as of 2013 has decreased pretty significantly. So at one point in time, low, moderate, and high invasions were equally shared. Most of the invasion or most of the Melaleuca dominated sites are now low Melaleuca abundance. So this is again a major accomplishment and continued success because of multi-agency cooperation and utilizing methods including chemical, mechanical, and biological control for an overall integrated pest management success story. So we moved then and utilized the framework from this Tame Melaleuca project in the Comprehensive Everglades Restoration Plan. So it's approximately the same footprint as we had before with the Tame Melaleuca, except that we're now including the Kissimmee chain of rivers and lakes. And it is tentatively entitled the Melaleuca Eradication and Other Exotic Plants Project. And again, this comes under that idea that a restored Everglades does not include invasive exotic plants. This is a partnership with the U.S. Army Corps of Engineers, the South Florida Water Management District as supporters, and then the Department of Interior and the USDA ARS as providers of resources, either travel resources or in the case of ARS, land resources to mass rear and release biological control insects. So the idea then is that we can conduct host range tests in this federal quarantine facility. We can ensure host range specificity, that the insects are going to be, a good utilization and be effective in the field. And then once we have the permit to release, we can then immediately move them over to the adjacent mass rearing annex that was completed in 2013. This is officially the first completed project of the Comprehensive Everglades Restoration Plan. And we are working hard to make sure that we are still on top of our plans and goals. This facility houses 4 offices, 3 postdocs, and then 2 large labs that focus on mass rearing 4 insect arthropod combinations. So right now we're focused on another wetlands invader, so sort of an ecotone invader, but really problematic old world climbing fern, as well as an aquatic macrophyte, water hyacinth, which everyone knows, and then also a sort of uplands invader, air potato. So these are accomplished again by hardworking folks who literally day in and day out rear insects and release them. And since doing so, we have annual meetings with our stakeholders to make sure that we're meeting benchmarks and that the goals that are set forth in the proposal and in the plan of action are still relevant. And that's one of the things that's made this successful. So we focused on these 3 species, and we've released over 2 million insects for controlling Lygodium microphyllum, water hyacinth, and air potato. Just to give you some successes from these efforts and from SERP in terms of our portion of SERP, we've had significant progress towards control and maintenance with Lilioceras cheni, this cute little red beetle, on air potato vine. So we've pretty much cut back recruitment from their aerial tubers, and height has been reduced, coverage has been reduced, and we're really entering a point where maintenance is feasible, especially if there's other intervention controls such as chemical spraying. The Lygodium is a different story. We're still sort of in its infancy, but this little brown moth has been seen, and we've had several more events like the one that's pictured here on the left, where we have massive population explosions where millions of individuals are flying around laying eggs, and their caterpillars are consuming massive quantities of foliage. This is an old hammock that was overtaken by Melaleuca at the Loxahatchee National Wildlife Refuge. These dead sticks are old Melaleuca trees that have been treated, so they were killed by herbicide, and then came in a secondary invader, old world climbing fern. So this is an insidious invader. It's spread by spores. It can pretty much travel anywhere in Florida, and it's particularly bad in wildland areas. That said, all of this brown is areas in which this moth and its caterpillar have been consuming. So since we started SERP, we've seen far more of these population explosion events. So again, more work needs to be done, but we're considering this a promising sign. In terms of the world's worst weed on Icornia crassipes or water hyacinth, we've had some good successes in researching and developing IPM methods for utilizing the weevils that are out there now and have been out there for the last 30 years, as well as a new insect from South America, Megamelus scutularis, in addition to reduced rates of 2,4-D and herbicide. We've come up with some novel methods for surveying the populations, and we've worked with the U.S. Army Corps of Engineers. So again, collaboration, utilizing their unmanned aerial vehicles or drones to release these insects in places that are difficult for us to get out. So again, we're continuing that interagency cooperation in order to move towards a common goal. We've expanded biological control along the Kissimmee River drainage, and we've been able to see our biological control insects, particularly Neomucetema conspercatalis, so that Lygodium moth, as well as Megamela scutularis on water hyacinth. So adding the Kissimmee River drainage, we've been able to increase the total range of the biological control organisms throughout that region and the larger SERP area. We would like to continue this momentum into future projects. As I said, air potato is a success at this point in the game, and we'll probably be moving 1 or 2 projects from full mass rearing and release to other projects. One of those is Brazilian pepper or shyness. So it's got these bright pink berries. It's sometimes called pink peppercorns or Florida holly. It is one of the worst weeds in South Florida and had experienced a 16% increase, at least in what we could survey between 2010 and 2012. Like Lygodium, it proliferates in remote areas, including this Cape Sable area out here, which is not any place that anyone ever travels to, but because these seeds are eaten by birds and by other seed predators, they get carried away to far remote places. It's very woody and brushy and impedes wildlife corridors and the ability of wildlife to move freely within areas. It infests more area than any other invasive plant in Florida. It's just big and burly, and it's difficult to control. If you ever travel in Florida all up and down the I-95 and Turnpike, you'll just see it along the roads. And again, it's not just along the roads, it's in some of our most remote wildlands. And in addition to Florida, it's problematic in Texas and Hawaii. Again, this is a plant with a wide ecological envelope. It can handle inundation, it can handle slightly saline, it can handle drying. So we're talking about something that is difficult to get rid of. We have 2 potential insects currently for the control of Brazilian pepper. One of these is Califia terebinthifolius, and this organism and its congeners, which are still in quarantine, are really host specific, and they form these galls, so they're leaf gallers. Another insect that is in the pipeline and has been approved for release, so Califia has been approved for release by the Technical Advisory Group, is Pseudophillothrips itchini. So this is the Brazilian pepper thrips, and it attacks the tips and growing tips of Brazilian pepper. Both of these were petitioned to tag in 2014, and both were recommended for release in 2016. And so if we can utilize the same framework as we did, we know that we have, for the previous projects, we know that we have established relationships with both land managers and cooperators and supporters to perpetuate these efforts indefinitely. Our cooperation with the University of Florida allows us to communicate readily with the public. In terms of Florida and the DOI and releasing in Everglades National Park, we're able to readily obtain permits to release in both the park as well as Big Cypress National Preserve. And this promotes a general idea of the safety and efficacy and acceptance of biological control. We've demonstrated the safety and the usefulness of integrating it with other management techniques. And then it also provides unique research and funding opportunities. You know, I'm a community ecology scientist. Looking at species interactions in these novel systems provides some really interesting research. There are other aspects, though, of the interagency cooperations and interactions that are not about boots on the ground getting rid of invasive species. One of these is importing insects. Remember that these plants are not native to North America. They come from other places, and so their natural enemies come from other places as well. And we conduct foreign exploration in those places with our collaborators, especially the Australians. Those insects are imported into US-based quarantines, and those are, that process is governed by the APHIS, USDA Animal Plant Health Inspection Service, as well as US Fish and Wildlife Service. Host range testing is conducted in the quarantine facilities by universities as well as ARS. We then put everything together for the technical advisory group, which is governed by APHIS, but is made up of multiple individuals from multiple stakeholding agencies and groups, including tribal organizations. And then US Fish and Wildlife Service will conduct the Invasive Species Act compliance, Endangered Species Act compliance, and APHIS will conduct NEPA compliance. And then finally, a permit is issued. So you can see that throughout this entire process, there are regulatory interactions as well as research collaborative interactions before an insect even ever gets released. Just to sort of give you an idea of some of the places where we have interactions and where there's potential for confusion, is that at the point of importation, we are there is shared regulatory oversight. So we are looked at by both USDA APHIS, who provides an importation permit and does the inspections at the port of entry, but all wildlife entering into the United States has to go through a US Fish and Wildlife Service law enforcement inspection at the port of entry. So those are governed under, two totally different mandates. Complete host range tests, like I said before, are evaluated by the technical advisory group for biological control agents of weeds. And again, this includes members of universities, of US Fish and Wildlife Service, of APHIS, of tribal groups who could be affected. So the TAG group is supposed to be really a group of stakeholders that can evaluate the data and the merit for a specific petition. The environmental assessment is conducted by both US Fish and Wildlife Service as well as APHIS. And then finally, the permit for release is issued by APHIS. One thing that we've run into is that because of several issues dating back several years, and I won't get into those, we haven't had any insects that have made it through the entire process of regulatory oversight. I'll just use the two that we're concerned about for Brazilian pepper. The Califia and the Pseudophilothrip itchini have both been recommended for release by the TAG group. There is a dual effort to complete the environmental assessment with US Fish and Wildlife Service, who conducts the Section 7 compliance and investigations, and then APHIS, which will do the public comment and the NEPA compliance. Because of some issues, none of these have been completed since 2012. That is actually not true. Well, it's sort of true. None have actually been completely completed, but I will say that Califia has now completed the Section 7 from US Fish and Wildlife Service and will just be starting its NEPA compliance with APHIS. So look for that for the public commenting period that should open fairly soon. But how do we make sure that we are being safe, make sure that these tests are robust, but also that we are able to utilize the research and the efforts that have gone into making sure that these are safe organisms for biological control? Do we need to increase communication? Do we need to have annual meetings between the regulatory agencies and petitioners? Does there need to be an increase in transparency where we can follow, much like you follow a FedEx package from point A to point B, that we can follow the petition from the original petition date all the way through, and have folks that are conducting these compliance investigations check boxes to say what's complete, what's not complete, and what's needed. This is an expensive process, a human expensive process. Can we talk about cost sharing between the petitioning agencies, the permitting agency, APHIS, as well as the investigating agency, U.S. Fish and Wildlife Service? And then finally, a question, again, for petitioners, and without risking seeming like this is a pay to play, but should there be a fee attached to filing a petition? Again, questions for other meetings and other places, but hopefully the ball is now rolling on these, and I would really like to thank those that are starting that ball rolling again after so many years. This is a really important tool to use. Selfishly, it's a really important tool to use in South Florida, where our wildland weeds are massive problems. But I know in other places, especially the West and the Northeast, the Mid-Atlantic, really everywhere in North America, we're combating these issues. And this is can be an effective tool, as I've shown before. So just to close, long-term interagency goals can help us, and long-term interagency interaction and collaborations help us to conserve the ecological function in communities and to reduce their level of invasiveness. It allows us to cooperatively manage lands that are adjacent, and especially watersheds where we have water and other resources, animals, insects, plants, seeds, moving from one place into another. As resources become more limited, having interagency cooperations is a good use of those resources and promotes efficiencies. And then finally, if we can duplicate the efficacy and the success of these previous integrated management projects, particularly like Tame Melaleuca or SERP, which we've been really successful to heretofore, we can make big strides in reducing costs and management costs in aquatic, wetland, and terrestrial communities. And the previous webinar had two questions, and I will address both of those. The first question was regarding international collaborations. So I've talked about our state and federal collaborations, but we do have international collaborations. So we work extensively with the CSIRO in Australia, which is essentially their research organization, and our collaborators there do much of the foreign exploration. And if we have plants that are from Australia, so I talked about Melaleuca, which is from Australia, we have another example, earleaf acacia, which we're just starting on. So they can do the initial surveys, they can even do the initial host range tests to screen out generalists before they ever even come over here. So it takes a huge amount of workload off of us and really streamlines the process. We also work with the Chinese Academy of Sciences and the Wuhan Botanical Garden in China. We've worked extensively with Fudei in South America to get the Megamelus scutularis insect out. So yes, we're a science organization and a science agency at the Agricultural Research Service. So our international collaborations are part of what makes this a successful endeavor. The other question I got was regarding the use of gene editing technology, or CRISPR, to try and develop biological control organisms that only last one generation. And so it would basically be like a bioherbicide. That's not really what our goal is with biological control. Our goal with biological control, as I mentioned before, is to have indefinitely perpetuating populations of these monophagous, folivorous insects that can last in perpetuity without any manipulation. So of course, nothing is inherently without risk, but we try to mitigate that risk as much as possible in the initial host range test portion of our work. So it's really, it would never be our goal to create an organism that only lasts one generation. Rather, we try to utilize the long established, long developed evolutionary relationships between specialist herbivores and their host plants. So with that, again, I'd like to thank our supporters, U.S. Army Corps of Engineers, South Florida Water Management Agency, Florida Fish and Wildlife Conservation Commission, as well as our many cooperators throughout the state of Florida and the federal government. So thank you very much.